Hemoglobin. Hemoglobin is a respiratory pigment present in the blood. It is a chromoprotein, conjugated protein and globular protein. Hemoglobin occurs in all the vertebrates. It occurs in the blood corpuscles of vertebrates and plasma of invertebrates. The invertebrate hemoglobin is also called erythrocronion. The hemoglobin is also found in the muscles of birds and mammals. The hemoglobin that is present in the muscles is called as myoglobin. It helps to store oxygen temporarily in the muscles. In uric acid, hemoglobin occurs in the nerve cells. It has a molecular weight of 68,000. Hemoglobin is a chromoprotein, that is conjugated protein. In a normal person, each 100 ml of blood contains 14 to 15 grams of hemoglobin. The total hemoglobin content is approximately 700 grams in a man. It is composed of two components, namely a protein component called globulin, that is 96%, and a non-protein component, that is a prosthetic group called heme, 4%. Globin is formed of 4 polypeptide chains. Of the other 4 polypeptide chains, 2 are identical, are called alpha chains. An alpha chain is formed of 141 amino acids. The other 2 identical chains are called beta chains. Each beta chain contains 146 amino acids. So hemoglobin contains a total of 574 amino acids. The heme is a non-protein and it is formed of iron and porphyrin. The iron is present in the fibrous condition, that is Fe2+, ferrous condition. The porphyrin is the pigment and it gives red color to the hemoglobin. There are 4 heme molecules in one hemoglobin. Hemoglobin combined with oxygen to form oxyhemoglobin. This combination is loose at low oxygen pressure. It leaves oxygen forming reduced hemoglobin. One hemoglobin molecule can bind with 4 molecules of oxygen. When hemoglobin is treated with hydrochloric acid, it forms a brown colored pigment called acid hemoglobin. When hemoglobin is treated with glass lactic acid, it forms hemine crystals. When the RBCs are destroyed, the hemoglobin is destroyed. This process occurs in spleen, bone marrow, and during the destruction, heme is separated from globin. The heme undergoes further breakdown. The iron portion is returned to the bone marrow to be used again in the manufacture of new hemoglobin. A large proportion of the heme is converted into bile pigments, that is bilirubin, and is excreted by the liver into the intestine. Some of it is reabsorbed and excreted in the urine as urobilinogen. Major portion of it is excreted in the feces as stercobilinogen and stercobilin, which are responsible for the brown color of the stool. Cytochromes. Cytochromes are heme proteins serving as an electron carrier in respiration and protein synthesis. Cytochromes are quite unusual enzymes and react with other enzymes rather than substrates of low molecular weight. Cytochromes were discovered by McMahon in 1886. The name cytochrome was coined by David Kelley in 1925. Cytochromes are present in all the cells of animals and plants and aerobic microorganisms. They are present mostly in the mitochondria of cells. They are called heme protein or hemoproteins because they contain iron. Cytochromes are globular conjugated proteins. Each cytochrome molecule has a protein part linked to a non-protein part. The non-protein part is called prosthetic group. The protein part is formed of single polypeptide chain of 104 amino acids. The non-protein prosthetic group is iron. The iron exists in the ferrous or ferric form, depending upon the state of reduction or oxidation. Cytochromes contain a colored pigment called porphyrin. Hence, cytochromes are chromoproteins. The cytochromes are generally red. There are 5 main types of cytochromes. They are named by the alphabets A, A3, B, C and C1. They are named based on their light absorption spectrum. Cytochrome A absorbs the longest wavelength of 580 to 590 millimicron. Cytochrome B absorbs slightly lesser wavelength, that is 556 to 558. And cytochrome C absorbs at least wavelength 441 to 550. The various cytochromes differs from each other in the nature of the prosthetic group and its mode of attachment to the protein part. The prosthetic group of cytochrome is heme. This is made up of iron and 4,5. Cytochrome C is most extensively studied. It is a conjugated chromoprotein existing in two forms, namely cytochrome C and C1. It is a small protein and the molecular weight varies from 12,400 to 13,000. It has an absorption spectra from 441 to 550. It is a basic protein containing a single polypeptide chain made up of 104 amino acids. Cytochrome C is a heme protein. The iron 4,5 prosthetic group of cytochrome C is heme. The heme C is covalently attached to the single polypeptide chain. The amino group of N-terminal amino acids is isolated in all the known cytochrome C. It is spherical in shape. The heme group is surrounded by many tightly packed hydrophobic side chains. The iron atom of cytochrome C is connected to the nitrogen atom of the histidine residue on one side and to the sulfur atom of the methionine on the other side. The 4,5 ring is linked by primary valency bonds to the protein through SH groups of two cysteine side chains. The resulting thioester bonds are very stable. In the electron transport chain, cytochrome C occupies a position just after cytochrome C1. Cytochrome C has great evolutionary significance. It represents an ancient protein about 1500 million years old and is of universal occurrence in eukaryotic organisms. Its universality is so conspicuous that cytochrome C from any organism could react in vitro with the cytochrome oxidase of any other organisms. Besides, the redox potential of all cytochrome C known so far is very similar. Finally, cytochrome C resembles cytochrome C2 and cytochrome C550 from bacterial cell wall. Cytochromes play important roles in electron transport system or respiratory chain. The 5 types of cytochromes are arranged in succession as an integrated chain in the mitochondrial membrane. They function as carriers, transferring electrons from different substrate to oxygen to form H2. Myoglobin. Myoglobin is an oxygen transporting protein present in the muscles. It serves as reserve store of oxygen in the muscle. It is found in skeletal muscle cells and is abundantly present in the dividing mammals including walrus, seal and whale. Storage of oxygen by muscle, myoglobin permits these animals to remain submerged for long periods. Myoglobin was extensively studied by John Kenrive. It is a water-soluble globular conjugated protein. It has a protein called globulin linked to non-protein heme group. It is a chromoprotein because it contains a red pigment. Myoglobin is made up of two components, a protein part called apoprotein and non-protein part called prosthetic group. The apoprotein of myoglobin is called globin. It contains of single polypeptide chain, but hemoglobin has 4 polypeptide chains. The prosthetic group of myoglobin is called heme group. The heme group consists of two components, namely an iron atom and an organic pigment called protoporphyrin. The protoporphyrin is made up of 4 pyrrole rings. The 4 pyrroles are linked in methane bridges to form a tetrapyrrole ring. 4 methyl, 2 vinyl and 2 propanyl side chains are attached to the tetrapyrrole ring. These substituents can be arranged in 15 different ways. Only one of these isomers, called protoporphyrin 9, is present in the biological system. The iron atom binds to the 4 nitrogens in the center of the porphyrin ring. The iron can form 2 additional bonds, one on either side chain of heme plan. These bonding sites are termed as 5th and 6th coordination position. The iron atom in the heme can be in the ferrous, that is Fe2+, or the ferric Fe3+, or oxidation state. The corresponding forms of myoglobin called ferromyoglobin and ferrimyoglobin respectively. Only ferromyoglobin can bind oxygen. Myoglobin can be crystallized. Myoglobin has a single polypeptide chain of 153 amino acid residues. The molecular weight of myoglobin is 17,000, about 1/4 to that of hemoglobin. Myoglobin has a compact shape. The single polypeptide chain is folded in 3 dimensions to produce a tertiary structure. The backbone of myoglobin is made up of 8 relatively straight segments interrupted by bends. Each straight segment consists of a length of alpha helix, the longest having 23 amino acid residues, and the shortest has only 7 amino acid residues. All the alpha helix segments have been identified as right-handed. Almost 80% of the amino acids in the myoglobin molecule are in the alpha helical regions, and the remaining 20% are in the bends. The inside of myoglobin consists almost exclusively of non-polar residues, whereas the outside contains both polar and non-polar residues. The tertiary structure of myoglobin is identical to that of the single alpha and beta chains of hemoglobin. Myoglobin binds with oxygen to form oxymyoglobin. Myoglobin has a much higher affinity for oxygen than hemoglobin. 50% of myoglobin is saturated at 1 to 2 mm HgO2 pressure. At the same time, 50% of saturation of hemoglobin requires 26 mm HgO2 pressure. The next topic is silk. Silk is a pasty secretion of silkworm around it. Silkworm is the caterpillar of silk moth, Bombyx mori. The silk is secreted by pair of silk glands, the modified salivary glands. The caterpillar secretes silk fiber on the 42nd day. It goes on spinning silk around it for 3 to 5 days to produce a silky coat called cocoon. The cocoon is made up of a single continuous thread. Silk is produced commercially by sericulture, where silkworm is reared for the production of silk on a large scale. They have tensile strength of about 3,193.27 kg per square centimeter, or nearly as great as steel. The diameter of silk fiber ranges from 0.046 mm to 0.083 mm. Chemically, silk thread is made up of a fibrous protein insoluble in water. Silk is made up of 2 types of insoluble proteins, namely fibroin and sericin. Fibroin constitute 80% and sericin constitute 20%. The silk thread has an inner core of fibroin surrounded by sericin. The silk fibroin is made up of 2 fibers and are held together or surrounded by sericin. The molecular formula of fibroin is C30H46N10O12. The molecular formula of sericin is C30H40N10O16. The fibroin of silk is a beta keratin. Fibroin are stringy, insoluble protein, but they are flexible and supple. They do not stretch. Silk fibroin and other beta keratin have a very high content of glycine and alanine, the amino acids with the smallest R group. Indeed, in silk fibroin, every other amino acid is glycine. The fibroin is made up of polypeptide chain. The polypeptide chain in silk fibroin exhibit beta conformation. In the beta conformation, polypeptide chain is extended into zigzag rather than the helical structure. The zigzag polypeptide chains are arranged side by side to form structure resembling as a series of pleats. Such a structure is called a pleated sheet. In the beta conformation, there are no interchain hydrogen bond. Instead, there are interchain hydrogen bonds between the adjacent polypeptide chains. In silk fibroin, adjacent polypeptide chains are usually oriented in opposite or anti-parallel directions, with the amino acid terminal at opposite ends. Economic importance of silk. The raw silk is used in the manufacture of woven materials. The knitted fabrics are used for the preparation of garments, parachutes, parachute coats, finishing lines, sieve for flour mills, insulation coils for telephones and wireless reservoirs, tires of racing cars. Silk is used for stain, crepe silk, velvets, gloves, socks and stocks. Dyed and printed ornamented fabrics are used for saris, jackets, salwars and vapors. Wool. Wool is a fibrous protein. It is the soft hair of sheep, goats, llama, etc. It is a fibrous protein. The color of the wool may be white or grey or black in color. The wool is hygroscopic and is a bad conductor of heat. As it is a bad conductor of heat, more heat is produced when the wool is wet. It is elastic, durable and is not easily inflammable. It is remarkable for its physical strength. It has the remarkable property of regaining the original shape when pulled and has abrasive resistance. The diameter of wool fiber is 12 to 80 μm. It is covered by cuticle. The cuticle encloses a central medulla and a peripheral cortex. Chemically, the wool fiber is made up of alpha-keratin. It consists of a number of polypeptide chains. The polypeptide chains of wool are extremely long. The polypeptide chains of wool are rich in amino acids that have hydrophobic or water-insoluble amino acids such as phenylalanine, isoleucine, valine, methionine and alanine. The polypeptide chains of wool exhibit alpha helical conformation. The alpha helix is visualized as chain wound around a cylinder. The repeating units is a single turn of helix. The alpha helix is maintained through many intrachain hydrogen bonds. The hydrogen bond is formed between the peptide bonds or peptide groups of every first and fourth amino acid residues. Wool polypeptide chains also contain cysteine. The adjacent polypeptide chains are linked by disulfide bonds. In wool, the adjacent polypeptide chains are oriented in the parallel fashion with amino terminal ends lying side by side. In wool, the alpha helic polypeptide chains are twisted around each other to form a three-stranded supercoiled structures resembling ropes, each strand being an alpha helical coil. Collagens. Collagens are fibrous proteins. The term collagen is derived from Greek word meaning of produced glue. They are the major proteins of connective tissue. About 30% of total protein in the mammalian body is collagen. Collagen are abundant in skin, tendons, ligaments, bones and in the cornea of eye. Collagen are insoluble animal protein. Collagen fibers are exceedingly strong and highly resistant to strengthening and also resistant to digestion by proteolytic enzymes. They are converted into soluble gelatins by boiling in water, dilute acids or alkalis. Collagen is one of the longest known proteins. The strength of collagen fibers is remarkable. A load of at least 10 kg is needed to break a fiber of 1 mm diameter. Collagen is made up of recurring polypeptide subunits called tropocollagen arranged head to tail in parallel bundles. A molecule of tropocollagen is composed of 3 polypeptide chains. Each polypeptide chain first becomes helically coiled to form an alpha helix. The alpha helix may be visualized as a chain wrapped around the regular cylinder. The three helical strands then wound around each other to form a triple helix or superhelical cable. The three strands are hydrogen bonded to each other. The hydrogen bond stabilizes the triple helix. The collagens are unique in their high content of hydroxyprolines and hydroxylysine. Collagen has an unusual amino acids composition and sequences. The proportion of glycine is all a collagen molecules is nearly 1/3. The amino acid sequence of collagen is remarkably regular. Nearly every third residue is glycine. Moreover, the sequence glycine, proline, hydroxyproline recurs frequently. Collagen is synthesized by fibroblasts of connective tissues. Keratin. Keratin is a fibrous protein. It is the main component of hair, wool, feathers, nails, claws, quills, scales, horns, and outer layer of skins. There are two types of keratins, namely alpha keratin and beta keratins. Alpha keratins are the main component of hair, wool, feathers, etc., and much of the outer layers of skin. Beta keratins are found in silk and spider web. Keratins are remarkable for their strength. They are hard and are non-flexible. They are insoluble in water. The keratin is rich in hydrophobic amino acids such as phenylalanine, isoleucine, valine, methionine, and alanine. Keratin is formed of long polypeptide chains. The polypeptide chains of alpha keratin exhibit alpha helix. The alpha helix is visualized as a ribbon wound around a cylinder. Many such three strands arranged of the polypeptide chains of hair and held together tightly because of covalent cross linkages. Fibroin of silk is a beta keratin. It is insoluble in water but flexible. The polypeptide chains in silk, hair, and wool exist in beta conformation. The polypeptide chain is extended into zigzag rather than the helical structure. In beta keratins, adjacent polypeptide chains are usually oriented in opposite or anti-parallel directions. In alpha keratin, the polypeptide chains are oriented in the parallel fusion. Albumins. Albumins are simple globular proteins. Albus means white. The albumins include serum albumin of blood plasma, ovalbumin of egg white, lactalbumin of milk, myosin of muscle, leucosin of cereals, legumulin of legumes. Albumins are made up of single polypeptide chain containing 585 amino acids. Albumins are soluble in water. They are coagulated by heat. They are precipitated by an acid or salt. Molecular weight is about 66,248. Serum albumin. The most well-known type of albumin is the serum albumin in the blood. Serum albumin is the number one most abundant blood plasma protein and is produced in the liver and forms a large proportion of all plasma protein. The human version is human serum albumin and is normally constitutes about 60% of human plasma proteins. All other proteins present in the blood are referred collectively as globulins. Serum albumins are important in regulating blood volume by maintaining the osmotic pressure of the blood component. They also serve as carrier for molecules of low molecular weight solubility, including lipid soluble hormones, bile salts, bilirubin, free fatty acids, calcium, iron, and some drugs. Competition between drugs for albumin binding sites may cause drug interaction by interacting the free fraction of one of the drugs, thereby affecting potency. The specific types includes human serum albumin, bovine serum albumin, that is cattle serum albumin or B.Sc, often used in medical and molecular biology labs. Low albumin, that is hypoalbuminemia, may be caused by liver diseases, nephrotic syndrome, burns, protein losing enteropathy, malabsorption, malnutrition, late pregnancy, etc. High albinism is either caused by dehydration or defect. The normal range of human serum albumin in adult above 3 years is 3.5 to 5 grams per dl. For children less than 3 years of age, the normal range is border 2.5 to 5.5 grams per dl.